Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So we were discussing cell signaling and recently we started discussing about receptor tyrosine kinases, how the activation of receptor tyrosine kinases could result into different signaling pathways. So we learned about the few signaling pathways which are mediated by RTKs such as RAS-MAP kinase pathway, the efferent signaling, and today we are going to learn about PI3 kinase signaling. Okay, so we discussed that this few different extracellular signal molecules, they can result into activation of different signaling cascades, but a same signaling cascade can be activated by different receptor proteins, right? So this is also a case. So we will for sure have a lecture in future where we are going to discuss about the crosstalk or a wide diverse network of different receptor proteins, different extracellular molecules, and how they are resulting into the different signaling pathways and how those signaling pathways they are interconnected to each other. So we will discuss about this web of signaling pathways. So today what we are going to discuss, the PI3 kinase signaling, is very crucial because it is activated by both of the receptors, the GPCRs as well as RTKs. So this PI3 kinase signaling can be activated if the RTK is activated, or it can be also activated by the activation of GPCRs. Okay, so that is why PI3 kinase signaling is very important. So for that, let's first learn about what does PI3 kinase do. What is the job of PI3 kinase? And for that, let's break down the name of the PI3 kinase. So it is a kinase, means it is an enzyme which is going to phosphorylate certain substrate. And what is its substrate? The substrate is PI. And why does this 3 is here? So this 3 indicates that 3 phosphate groups are being added by the kinase enzyme over the PI. PI means phosphatidylinositol, is a phospholipid present in the plasma membrane. So you remember when we discussed about cell biology, the plasma membrane lectures, we discussed about phosphatidylinositol, which is a unique phospholipid because it has inositol ring, and inositol ring has multiple sites over which phosphate groups can be added. So before this, when on the phosphatidylinositol, phosphate groups are added. Okay, that results into formation of phosphoinositides. So those are known as phosphoinositides. So phosphoinositides we discussed in the plasma membrane lectures that they are very important, very crucial for the signaling to go on. And we have already discussed one kind of phosphoinositide, that is PI4,5 bisphosphate. So we learned how this PIP2 it can be either cleaved by phospholipase C beta, or it can be cleaved by phospholipase C gamma, which results into the formation of IP3, which is the intracellular signaling molecule, and diacylglycerol, which is lipid soluble secondary messenger. So we learned about this PIP2. So today we will be learning about the signaling which is done by PIP3. Okay, so PIP3. Let me just write it something like this. Phosphatid PIP3 that is going to do the signaling. Today we are going to discuss that. It is the phosphatidylinositol 3, 4, 5 trisphosphate. Okay, so we are going to discuss the signaling which is done by this molecule. And how does this molecule is formed? So this molecule is formed by the job of PI3 kinase. So on the phosphatidylinositol, PI3 kinase works and it results into formation of PIP3, PI phosphatidylinositol 3, 4, 5 trisphosphate. Okay, so this PIP3, this molecule serves as a docking site for multiple intracellular signaling molecules, and they do their own downstream job. So we learned that cellular signaling is very important, so that two cells they are able to communicate with each other, right? But signaling is also crucial for the cellular events such as cell division, cell apoptosis, and cell survival. So the PI3 kinase signaling, it is crucial for cell survival, and that is what we are going to discuss today. So due to the activation of PI3 kinase, some signaling pathways which are referred as AKT-mTOR pathway, they are activated and they result in the cell survival. We will be discussing about AKT-mTOR pathway in next lecture. Today we are going to only discuss how PI3 kinase, it when it gets activated by the receptor tyrosine kinases, it activates AKT, which is going to do its downstream job, which will result into the survival of a cell. Right, so that is the purpose of the discussion today. We are going to only learn about the how PI3 kinase, it is going to phosphorylated phosphatidyl inositol, result into the formation of PIP3, and how PIP3 is going to result into the activation of certain molecules which are crucial for cell survival. Okay, that's the thing. So this phosphatidylinositol could be phosphorylated on multiple sites. We have discussed about PIP2. Today we'll be discussing about PIP3. So the PI3 kinase, there are different categories of PI3 kinase depending upon what type of cellular effects that this kinase have. So PI3 kinase is categorized into three different categories due to the because it results into different cellular effects. The first category is referred as class 1, second category is class 2, and the third category is obviously class 3. So these are three different categories of PI3 kinase, and all of those different types of PI3 kinase, they are going to do different jobs. The class 1 PI3 kinases, they are also subcategorized into two types: the class 1A PI3 kinases and class 1B PI3 kinases. The types of class 1A PI3 kinases is P110 alpha, P110 beta, and P110 delta. While the P110 gamma is the class 1B PI3 kinase. The class 2 PI3 kinases are PI3 kinase C2 alpha, PI3 kinase C2 beta, and PI3 kinase C2 gamma. So these are the different types of class 2 PI3 kinases. The class 3 PI, on the contrary, class 3 PI3 kinase has VPS34 as an example. So all of them, they are going to result into different results. So the class 1 results into cell survival, class 1 PI3 kinases, and that we are going to discuss today. How this class 1 category of PI3 kinases, they are going to do the job of cell survival. On the contrary, the class 2 kinases, they are very important and they help in vesicle trafficking and the clathrin mediated endocytosis. They do the clathrin mediated endocytosis. While the VPS34, they are important in autophagy and nutrient regulation. So this is how all of those different types of PI3 kinases have different jobs. Okay, so the class 1 category, the class 1A category of the PI3 kinases, it is being activated by receptor tyrosine kinases. So this class 1A, those PI3 kinases, they are activated by receptor tyrosine kinases, whereas the class 1B kinases, they are activated by GPCRs. Okay, so today we are going to discuss about the RTK mediated activation of PI3 kinase. But let's also discuss briefly about GPCR, how GPCR activates the receptor tyrosine sorry, not the PI3 kinase. Okay, so I hope you remember that when we discussed about the GPCRs, how the different types of G proteins, when they are being coupled with the 7' transmembrane protein, the downstream effects of of the signaling pathway changes, right? So if the receptor protein, the G protein coupled receptor, it is being coupled with a GQ protein. Okay, so this GQ protein, when it splits into the alpha subunit and a beta gamma subunit, G alpha subunit is going to have its own downstream effect, which is going to result into cytoskeletal movement, right? We discussed that. But beta gamma also has some downstream effects. So one of them is that G beta gamma, it activates P110 gamma, okay, which is a phospholipase, not phospholipase, which is a PI3 kinase, which belongs to the category 1B, right? So this belongs to class 1B, right? So this class 1B PI3 kinase, this is being activated by a GPCR, the GQ protein, by the beta gamma subunit, and the RTKs. Okay, RTK, they activate the class 1A, class 1A PI3 kinases. So let's learn in detail how the RTK do the activation of those PI3 kinases. So this is the plasma membrane, this is the exoplasmic leaflet, and this is the cytoplasmic leaflet. Right, so so just like other RTKs, those the RTKs which are going to activate PI3 kinases, they also have similar functioning. So the receptor tyrosine kinases, they get coupled, they get dimerized on the arrival of the extracellular signal. So as I say that PI3 kinases, they are important for the cell survival. And so just guess, take a guess that which kind of extracellular molecule is going to do the job here. Obviously a growth factor, because growth factors are the ones which are going to result into the cell growth, cell survival, or they could also result in cell division. Cell growth is of two types: either cell is multiplying, cell number is increasing, or the cell cytoplasm, so the cell matrix, whatever cell contains are there, they are increased. So cell is either growing in size or it is growing in number. So that is what we refer as cell growth. And cell growth is it is signal, when a signal growth factor arrives, then the cell growth happens. So the signal that will arrive and result into PI3 kinase activation, that will be the insulin like growth factors. So insulin like growth factors are the one which are going to result into the PI3 kinase signaling. So when insulin like growth factors, they bind to the receptor. So now this receptor is receiving insulin-like growth factor, right? So it can be named as insulin-like growth factor receptor. So when this receptor binds with insulin-like growth factor, IGF, now this will get dimerized. And due to the dimerization of, so this receptor is a receptor tyrosine kinase. It means that the intracellular domain of this receptor has intrinsic tyrosine kinase activity. So those green ones are the tyrosine kinase domains which are present intracellularly on the cytosolic side, right? So due to the dimerization, those tyrosine kinases will get activated and they will result into the addition of phosphate groups over the tyrosine residues. So those addition of phosphate group will result into phosphotyrosine residues. And due to the formation of those phosphotyrosine residue, now a PI3 kinase can come and bind to this phosphotyrosine residues. And due to this binding of PI3 kinase to those phosphotyrosine residue, this PI3 kinase will get activated. Okay, so this PI3 kinase binds to this phosphotyrosine residue. There are two cases for the activation of the PI3 kinase. One is that when this RTK is getting activated, as I said, that PI3 kinase will directly bind itself to the phosphotyrosine residue. So PI3 kinase has a subunit known as P53, which is a regulatory subunit. Okay, Means if the subunit is bound to the phosphotyrosine residue, that is going to regulate the activity of PI3 kinase. But it means it is going to activate the PI3 kinase. So this P85, this regulatory subunit, it binds to the phosphotyrosine residue. Okay, so phosphotyrosine residues of the activated RTK, and now the PI3 kinase will get activated. But the other way of activation of PI3 kinase is that instead of adhering PI3 kinase directly to this receptor tyrosine kinase, some adapter protein attach themselves to those phosphotyrosine residues. So SH2 domain containing adapter proteins such as GRB2 or GAB1. Okay, those they are the ones which are going to adhere to the phosphotyrosine residue, and over those adapter proteins, the PI3 kinase attaches itself and it gets activated and does its downstream job. So there are two ways to get to activate the PI3 kinase. Either PI3 kinase itself directly attaches to the phosphotyrosine residue, or it gets attached to the phosphotyrosine residue with the help of some adapter proteins such as GRB2 or GAB1. Okay, so these are two different ways of the PI3 kinase activation by the RTKs. Okay, so we are going to learn about this first phenomena. So this PI3 kinase, okay, this is the PI3 kinase, and this particular domain which is binding to this phosphotyrosine residue is P85. Okay, and this is PI3 kinase, which is now activated due to the binding due to the binding to the phosphotyrosine residue, to the activated RTK. So now this activated PI3 kinase, it will do its job. So it is a kinase, so it is going to phosphorylate PIP2, which is already present over the cytoplasmic leaflet. So over the cytoplasmic leaflet, we discussed that PIP2, this molecule, is adhered with the help of lipid anchor. So let me just draw PIP2 over here. Okay, let me just extend this cytoplasmic leaflet, and here, with the help of some lipid anchors, PIP2 is adhered. So you remember, so you already know that PIP2 is here because we have discussed this in the previous classes of the signaling, right? So to this PIP2, so I will just write it here. So PIP2 will be converted into PIP3, or let me write it as PI345 trisphosphate. Okay, so this will be done by PI3 kinase. So once this PIP3 molecule is formed, this PIP3 molecule is very crucial because it is going to serve as a docking site for certain intracellular molecules. So this PIP3 molecule, to this PIP3 molecule, two different types of enzymes can attach themselves. The very first enzyme that is very crucial for cell survival is AKT, and other enzyme which gets bound to this PIP3 is known as PDK. Okay, so both of them, they can attach themselves to PIP3 because both of them has a particular domain, a particular domain which is known as pH domain. Okay, both of those protein has pH domain. So what is pH domain? pH domain is also, it is the plextrin homology domain. So multiple proteins have this domain, and this is actually sorry, this pH domain is actually a protein-protein interaction domain. So you remember that there exists some protein DNA interaction domains in the proteins, so that the protein could adhere itself to a DNA molecule, right? So this pH domain is a protein-protein interaction domain. Okay. But this protein-protein interaction domain containing molecules, they have affinity to the PIP3 molecule. So this increases the specificity. So those pH domain containing molecules such as AKT and PDK, they can bind themselves to the some pH domain affinity to the proteins which have pH domain affinity. So let's say that this protein It has a pH domain. Okay. So the way that PIP3 has the pH domain affinity, some other protein could also have pH domain affinity. So let's say that this protein, this particular domain, this protein has a subunit which has affinity. So this violet has affinity to the pH domain. Okay, so due to which this domain containing proteins, they are able to adhere themselves to the pH domain containing proteins, right? And that is why this is the reason why not all PIP3 they are occupied with this AKT and PDK, because the pH domain, not all pH domain containing proteins, they are adhering themselves to PIP3, but the presence of this pH domain in the AKT and PDK makes them highly specific to PIP3, because PIP3 can interact with this pH domain. So note down this point, that PIP3 has the capability to interact with the pH domain. So pH domain, it was named like plextrine homology, because this domain was first identified in the platelet protein plextrine. So that is why it was named simply plextrine homology domain. Okay, so this pH domain is present in both AKT and PDK, and that is why both AKT and PDK, they will adhere themselves to PIP3, and they can do their own downstream signaling. So this AKT, it is actually a serine threonine kinase, and that is important for cell survival. So this AKT is it is going to activate mTOR, okay, and that is going to result into cell survival. But how does this happen? That we'll discuss in next class, where we will discuss about AKT mTOR pathway in detail. But this AKT is just bound to PIP3. It is not activated yet. How does this get activated? This gets activated with the help of this PDK. So what is the full form of PDK? PDK is phosphatidyl inositol phosphatidyl inositol. No, not phosphatidyl inositol. Phosphatidyl inositide dependent protein kinase. Means this is a kinase which is going to phosphorylate some substrate which is dependent on phosphatidyl inositols. So this is phosphatidyl inositide, right? So this is dependent on PIP3, and that is why it is called as phosphatidyl inositide dependent protein kinase. So this is a kinase. So it phosphorylates this AKT. So this will phosphorylate AKT, and now this phosphorylated AKT is activated, and it is going to inhibit some molecules which will result into the activation of mTOR, okay, and that will result into cell survival. This pathway, we'll learn about this pathway in next lecture in detail. Okay, so let's now have a look at the schematic given in Bruce Alberts, so that the whole lecture will be summarized in one shot. Okay, so here you can see. So we discussed that both RTKs and GPCR, both of them can activate PI3 kinase. So we discussed about the activation of PI3 kinase with the help of receptor tyrosine kinases. So this receptor tyrosine kinase is a receptor for insulin-like growth factors. So when insulin-like growth factors, they arrive on the site, they dimerize this receptor tyrosine kinases, which activate tyrosine domain and does the phosphorylation of tyrosine residues present on this intracellular domain. So this creates the phosphotyrosine residue, to which the PI3 kinase can adhere itself. So the attachment of this PI3 kinase to this phosphotyrosine residue activates the PI3 kinase, and this PI3 kinase will add the phosphate group on the PIP2 and will convert the PIP2 into PIP3, the PI345 trisphosphate. So this PIP3, it can interact with the pH domains. So the pH domain containing proteins such as PDK1 and AKT, both of them can interact with PIP2s. Okay, so PIP2 will be occupied with AKT. Some PIP2s will be occupied with AKTs, and some will be occupied with PDK1. So the PDK1, which is being occupied by PIP2, Now this is activated and it will phosphorylate the AKT. And the phosphorylation of AKT activates this protein, and due to this activation, it results into the activation of some downstream proteins such as mTOR, which results into the cell survival. And how does it do the cell survival? That we'll discuss in the next class. So I conclude the lecture here. Till then, see you. Bye bye.